very good evening to the professor and all the members. So today I'm going to just brief my experimental work and what I have planning in this meeting to finish it. So first of all, I just go with my personal timetable, like what I'm going to do in this week. So my main focus is to do the two different kind of things. Like first one is like to on Wednesday, as a professor said, like we have to go to the SNU for the next plan for this. Like we just go with the LCR meter and to measure the functionalized P6 chip that who like the professor like Sangte Kim having now. So it is functionalized with the some samples. So we just have to measure it and go for the positive results, and do with the next step. And the second one is like what I'm doing as suggested by the professor. Like I have to using the PDMS based P6 chip to using our COVID, like SARS-CoV-2 antibody and antigen in our lab. So just check it out, like is it working or not. So this two is on this week, and the third one is like having the aptamer based biosensors. So these are the three different kind of things like I'm working on it. So as I said, like previously, like 2 weeks before, 2 to 3 weeks before, we have some sort of P6 chip to just look into our like you are having functionalized with the aptamer. There are two different kind of aptamer is immobilized from the Sangte Kim. So is having like N protein and S protein. So these are two different kind of protein that is the like 10 on 10 chips. So these are the various steps that we have to follow. Like first one is the cleaning surface, and then APTS functionalization using he used the glutaldehyde after that, and they just immobilize the aptamer N protein and S protein. So these are the two different kind of proteins that the Professor Sangte Kim used. And of course on Wednesday we just go there and we found that this kind of protein in the on the PDMS based chip. So these are the things that we have to look onto the on Wednesday. So but for this now we don't unfortunately this I just got this kind of chips for the AFM SEM FTIR, but due to some reasons it's already on hold. I don't know what is going on, but hopefully um, what professor said in the next things like to go with this chip or not. um, yeah. And it's I already given this 30 number of D5 chip to the Professor Sangte Kim, and on Wednesday hopefully we will get the chip and to make the measurements using the LCR meter. And also I given him the 22 chips P6 PDMS based chips for the next experiment. So the same thing also I suggest to like on Wednesday we will go with the LCR meter and to measure these chips. And yeah. So this one is my different task that I have currently. I have 25 P6 PDMS chips. Is a P6 chip obviously, and so I have to follow this step in a four different kind of approach. And I just go with that one. So it's like the, using P6 chip and look into it, like what we have now in our lab, like SARS-CoV-2 antibody antigen. So just double check like whether it is working or not. So in this, like from tomorrow onwards I will just go with this one as well. And also I just have planned to make the two different kind of composite, like graphene oxide composite, like reduced one and the without without reduced graphene oxide. So we just make sure like which one is better for our um, like consecutive steps, like how it can combine together. So this is the thing that I'm working on it in the from tomorrow onwards. And yeah, it's of course at the last end, like I just have to work on the review article as well. So I just implemented these five different kind of updated contents in our review paper, and hopefully it just complete into the next week. So still I'm just working on it. So these are the things that I'm working here. And obviously it's like it's a kind of a series that I have to working on it, like on Wednesday, just go with there, and if we got the positive results of this using P6 PDMS chips, so we just go through with using these chips and make more and more and more number of chips, and hopefully if this work is working well, then we just have to make a different kind of approach in our biosensor stream, like we just using PDMS fabrication, and then we use the bioanalytes to make the different kind of scenario, and hope for the best. Definitely we will go with this, and of course Professor Sangte came having the different kind of proteins, like having N protein and S protein. It's a different way to find out to make how to reusable biosensors. Yeah. So that's all, sir.